Hello, everyone. I'm Pablo. I'm a former master's student from Leiden University, and I will be presenting today my master's thesis project that I developed at the LMC in the biosemantics group on the topic of knowledge graphs and explainable AI for drug repurposing on rare diseases. Okay, so the rare disease that we will be focusing on is called Duchenne muscular dystrophy. It's an X-linked recessive disease affecting 1 in every 5,000 male births. It currently has no cure, and its life expectancy is of 21 to 40 years old. This disease is caused by mutations in the DMD gene, which interestingly is human largest gene with 2.4 billion base pairs. It codifies for dystrophin, which is a protein that serves as anchor between the muscle cells and the extracellular matrix. So what happens is that when this dystrophin is affected, it becomes unstable and it leads to fragile muscle cells. This way, most of the symptoms of the disease are muscle-related. So patients usually have weak muscles, cardiac deficiencies, digestive dysfunctions, and they usually end up dying of respiratory failure. So the objective we had with this project was to see if we could use AI methods to find drug candidates that, to treat the symptoms of rare diseases. But we didn't want to obtain just the drug candidates. We also wanted to make special emphasis in obtaining explanations that could help both researchers and clinicians to generate testable hypotheses. So here's a summary of the methodology that we've developed throughout this project. So there are 3 main blocks. First one, where we'll be building a knowledge graph containing information of the disease. An AI part, where we will be training a graph neural network model to obtain the predictions. And finally, an explainable AI part, where we will be using where we have used a tool known as GNNExplainer to obtain the explanations. Okay, so the first part is, as I said before, building the knowledge graph. And to do this, we have used 3 different data sources. Our main source of information was Monarch, from which we have obtained all the information regarding the genes, the symptoms, the phenotypes, anatomical structures. And to build the knowledge graph, we've used a tool known as the Knowledge Reviewer. However, Monarch does not specialize in drug information, so we had to complement our graph with 2 more data sources: Drug Central for drug target information and Therapeutic Target Database for drug disease information. In total, we built 2 graphs. A small graph that was used as a toy example to test our model, and a larger graph that contained more information. So the way Bioknowledge Reviewer builds the graph is that you give it a number of seeds, and it will create the graph based on the neighborhood of those seeds. So for the small graph, we used 2 seeds, one for Duchenne muscular dystrophy and one for DMD. And in total, we obtained a graph with more than 10,000 nodes and almost 100,000 relationships. For the larger graph, we also used these 2 seeds, but we also incorporated seeds for all the symptoms and phenotypes of the disease. And in total, the large graph had more than 80,000 nodes and almost 2 million relationships. Okay, the next part is to build the AI model. In this case, we have focused the problem as a link prediction task. So we would like to know if there is a potential link between a certain drug and a certain disease. And as I said before, we used graph neural networks. These were built with 2 libraries, PyTorch Geometric and Snap, or DeepSnap. And the idea, so the output of these models was to obtain a node embedding in such way that the dot product between 2 nodes should reflect the probability of a link existing between those nodes. So, for example, if we took the node embedding of A and the node embedding of B, we obtain the dot product and pass it through a sigmoid function, we would expect that value to be as close to 1 as possible because there is a link between those 2 nodes. And similarly, if we follow the same process for B and E, we should expect that value to be as close to 0 as possible, as there is no link between them. And for the input layer, we created several node features with a tool known as H2Vec, but due to time limitations, I won't enter into too much detail, but it's basically a modification of Node2Vec that it also takes into account edge types to create this node feature vector. And finally, there is the explainable AI part, where we used a modified version of GNNExplainer. And here's a little insight of how GNNExplainer works. So when you usually make a prediction, you obtain several probabilities. In our case, we have a certain probability that there is a link between two nodes and a certain probability that there is no link between those nodes. And what GNNExplainer does is that it is going to remove edges from our knowledge graph, and it's going to see how the probabilities are affected. So for example, if we remove these three nodes from our graph, we can see that the probabilities changes a lot. So this must mean that these three nodes are relevant, or our GNN is taking them into account to make its prediction. On the other hand, for example, if we remove these other three nodes, the probabilities remain more or less the same. And this must mean that our GNN model, these edges are irrelevant to our GNN model. And in the original paper of GNNExplainer, they implemented it for node classification and graph classification tasks. However, they also mentioned that it could be extended to link predictions. And in this project, we also implemented this version that is compatible with link prediction tasks. And here are some of the results. So here at the top, you can see the area under the ROT curve for both the small and the large graph, which is one of the standard performance metrics for link prediction tasks. And here at the bottom, there are also additional performance metrics such as precision recall and the F1 score, all of them above 90%. Next, we made an analysis a bit more in detail for the predictions. So for each symptom, we selected the 3 new drugs with the highest score, and then we searched in the literature to see if these associations had been reported. And in the case of the small graph, almost more than 20% of the predictions had supporting evidence, and in the large graph, a bit more, around 27%. And there were also cases where the drug was actually causing a certain symptom, and we labeled these interactions as contraindication evidence, and they were more or less the same in both graphs. Next, we also analyzed the explanation. In this case, we created explanations for 7 different symptoms, and we divided them into 2 groups, complete explanations and incomplete explanations. So complete explanations are those where there is a clear path connecting both the drug and the symptom. And incomplete ones are those where there is no such path. So we either have 2 separate clusters or a unique cluster, but where only one of the 2 components is present. So we only have either the drug or the disease or symptom. And here we regard complete explanations as the ones that are truly useful, as they can be easily interpreted. And here are some of the results. So when the model created a complete explanation, it had supporting evidence 62% of the time, in contrast with incomplete explanation. So when explanation was incomplete, the prediction only had supporting evidence 28% of the time. Here are also some results, this time looking at individual graphs. So in the small graph, it was quite interesting because if the prediction had supporting evidence, our model was always able to create a complete explanation. And similarly, in the large graph, when the prediction didn't have any supporting evidence, it was never able to create a complete explanation. So the explanation was always incomplete. And it's also worth noticing that the small graph tends to create more complete explanations than the large graph, where the explanations tend to be incomplete. However, a complete explanation does not necessarily have to be a good explanation, as the explanation might be a bit messy or it might don't have a biological sense. And for these reasons, we also perform an analysis, a manual literature search for the explanation to see if the explanation had been reported in the literature. So out of the 21 complete explanations we had, only one had been described in the literature. However, there were two drug symptom associations where researchers didn't know why a certain drug was causing a certain symptom. And we think that models like this may help clinicians or researchers to create or generate hypotheses. And finally, what you can see here is a table showing all the drugs that were suggested by our model, the number of times they appear, the same but expressed in percentage, and the number of times they had evidence or they were contraindicated for both graphs. And what you may notice at first is that there are some drugs that appear to be recommended for almost every symptom, and in fact don't have or have very few supporting evidence. And we label these drugs as promiscuous drugs, and we think of them as noise that appears in our model. Additionally, it was quite nice because there were some drugs that were that are currently undergoing a clinical trial to see if they can be used to treat Duchenne muscular dystrophy. And there were other drugs that they weren't undergoing a clinical trial, but they were of the same type of a drug that was undergoing a clinical trial. So, for example, here, methlosigine is a gram-negative antibiotic, and there is actually a gram-negative antibiotic that is being tested to treat symptoms of Duchenne muscular dystrophy. And here's here are some of the conclusions of our work. So the main contribution of this work is that we provided a model that clinicians and researchers can use to create explanations for drug candidates in rare diseases. And these explanations can be used also to generate testable hypotheses. Additionally, the AI model, the GNN model, had a really good performance, and often the drug candidates were supported by the literature. As I said before, we also implemented a version of GNNExplainer that makes it possible to work with link prediction tasks, and also with returns explanations that are human readable by using common biontologies. And for the near future, we would like to make the most of ontologies to improve both the quality of predictions and explanations. Also incorporate more information to our graphs, such by including maybe stuff like drug side effects. And also implement heterogeneous GNNs that are capable of extracting all the information from our knowledge graphs. And finally, I would like to thank both my supervisors, Katie and Nuria, and the whole biosemantics group, as they have been of incredible help and support throughout the whole project, and also all of the people that are attending here. And I hope you've enjoyed the presentation, and if you have any questions, I will gladly try to answer them.
So thank you very much. We have time for some questions and discussion.
Yeah, hi. Maybe repeat the question. But so I didn't quite understand the ontologies that we use. They have the benefit of those ontologies was that they were hierarchical. They had relationships in them, right? And then they could find those paths in the graph that were constructed from those. Right? Is that how? What's the value of the ontologies?
Mm-hmm. Okay, so the question is, what is the value of the ontologies? In particular, ontologies seem to have some relations, and where these relations used to construct the graph, or was there some additional semantics in these relations that was being used?
Okay, so I think that the ontologies become really important in the explanation part, as once the graph is constructed, thanks to ontologies, they can be easily interpreted by humans. What was the second question? How were they used in the building of the graph or something?
I think the question was, so I think the first part of the question was whether the ontologies were primarily used to build the graph, but the second part, I think the implied question was whether there was any of the semantics of these relations that is present in the ontologies used as part of the model, or was it only used to generate the graph?
They were only used to generate the graph. Yep.
Okay, thank you. You also had a question?
Yeah, so if he goes back to the table to this
Could you go back to the table, please?
Which one?
yes.
This table, the table of drugs.
Okay, this one.
So for the top one, the percentage is appearance 25. The percentage is 92%. And then the contradiction is 8. If they look at that, why it is happened this way? And then it seems like the good candidate will be the one who names who with contradiction as zeros. Is that true?
So Asia is asking, when looking at the first drug, there are 25 appearances with a 92% and 8 contraindications. So have you investigated why this would appear like this for this particular drug? And the second question is, would you identify drugs with 0 contraindications as the best suggested drug based on this method?
So why this drug was in fact the one that appears the most so it's the most suggested drug for the symptoms? I didn't make a lot of investigation on it. I assume that it's they are drugs that they have a really similar structure when looking at it from a similar neighborhood. So maybe they are not very specific and they tend to appear in all of the symptoms. And the second question, which was it? If I had seen a drug that had
0 contraindications, because this one has 8 contraindications.
So so here, for example, like the third one that had 0 contraindications, or what do you mean exactly?
Yes, would you identify good candidates when a drug has drugs that have 0 contraindications?
So for me, a good drug candidate would be one that has not few contraindications, but a lot of evidence. So I would regard this column as the one that I would look at it if I was looking for good candidate. I don't know if that answered the question.
Mm-hmm. Okay. Any other questions or comments? Please.
In your future work, do you intend for top-ranked drugs, for instance, to also include strength for their therapeutic effect? Drug strength.
So in your future work, are you considering to include drug strength? Strength.
So
yes.
So 10 milligrams works for one patient, but 5 doesn't work for the rest of the
yes.
work.
So would you also consider dosage or the strength of a drug in the future work?
Well, it's a really interesting idea, and I think it should be possible because maybe you can implement something in the actual link. You can always instead of using just a plain link, you can use weight links, and maybe in that way you could also incorporate information such as dosage, or also as an additional feature to drugs. Yeah, that's really a good idea. Thanks.
Mm-hmm. We had one more question.
So literature-based discovery work is something that has been going on for many, many years, right? And one of the best examples of that is SEMREP from the NLM, which is built on top of MetaMath. And there are a lot of published papers where they have discovered exactly this kind of novel use of drugs or mechanisms of action based on existing discoveries literature. So I'm curious to see how this compares with those other approaches that they've been doing.
Yes. So the question is, how does your work, this graph neural network, together with the explanation, compare to other approaches in the field? For example, SEMRAP, which is based on literature mining and developed by NLM, but there are other approaches as well. So how have you compared this, and how does it compare to other approaches?
So works like this that focus on rare diseases, to our knowledge, they weren't too many of them. I think that it's, and especially ones that provide explanations, so it's a bit hard to compare it to other works. So I don't have actually a good answer to that. There is, or as I said, to our knowledge, there aren't many works that are so specific.
Okay. One last question. Then
Yeah, there, it's kind of a follow-up question. So you can construct your graph based on ontology, right? Or you can construct a graph based on co-occurrences of entities you find in literature. Wondering what's the benefit to having one or the other?
Mm-hmm.
You know, you can argue eventually all the knowledge from literature should make its way into an ontology, or into a database. So is that maybe where like the literature is more on bleeding edge, kind of weaker evidence perhaps? This ontology is well-known fact.
okay, so we have another question. So you are building your graph based on an ontology, but the alternative could be to take, for example, co-mentions in literature to generate the graph, or other kinds of associations. So what is the benefit of using an ontology compared to using literature co-mentions?
Well, I would have to think a lot about it, or to think a little about it. Maybe Nuria might have a better answer than one I can give. But to me, the most important part of using ontology-based graphs is the fact that they more in the explanation part. So the fact that it provides explanations that can be easily read and understood both by humans and machines. So I think that would be the main advantage.
Okay, so thank you very much. Let's move on to the next presentation. Thank you again.
Thank you.